to have Angelo visit us from SISSA. Angelo has been working on chromatin, basically using polymer models to explain chromatin organization. And today he's going to talk about physics of entangled polymers from chromosomes territories to separates.
Thank you very much, Bud, especially for the invitation. I mean, I was when I received your invitation, I mean, I was very happy. So one thing that I would like to say is, if you, interrupt me whenever you want. I think it's a bit the purpose of this institution also to share ideas. So I don't mind being interrupted. So what I will present here today, in fact, oops, how can? Oh, okay. So it will be, as the title says, it will be about the modeling of chromosomes by using genetic polymer physics. But actually, I've also, if I have time, to talk about some recent work we did, basically using some kind of very similar physics that we use for chromosomes to understand the folding of the folding of RNA molecules. And also, if I have time, I don't know actually, but if I have time, I will also try to tell you about the recent field theoretical approach to interacting polymer solutions that we have just published. And because it's all related to my expertise, so I think it will be interesting, hopefully for you too. So before starting, I would just like to acknowledge, because this is common to all this work, some computational resources I got for doing all these works in the course of the years, and also a cost action, which was very useful. To share all this idea about these works during the last 4 years. Now the action is expired, but it was related precisely to the main topics of this talk. So the, let's say, topology in soft matter in general, and in polymers in particular. So the first part of the talk is actually a bit an old work, as you will see. So it's the last paper, I think it's 3-4 years ago, and it was mainly based on a long-time collaboration with Ralph Everett, which probably some of you know well. Actually started when I was a postdoc in his group in Dresden, and then we continued together to collaborate. But there were also very important inputs from all these other people, and I'm sure you recognize some of them, as Shura Grossberg, Michael Rubinstein, and Kurt Kremer, and also my friend and collaborator Jan Smrek in Vienna. So what was all about? I don't know why it's a bit slow. It's weird. I see how the thing's changing, but there no. I don't know. Maybe you just stop share. Yeah. Right. Okay. Was disconnected. Disconnected. Yeah, it's weird. Sorry. Okay, so now there's something. Yes. Yeah, it should work now. Okay, so yes, as I was saying, I was interested in the problem of chromosome folding, and actually there was the talk before, which was very useful as an introduction in this respect. So the idea is the following. Basically, very few elementary considerations. So we have that each human cell contains a very large amount of DNA. Actually, it's about 6 billion base pair of DNA, and if you if you unfold this DNA, it's about 2 meters inside the nucleus of each cell. Okay, this is partitioned into, let's say, some chromosome, some number of chromosomes, like 46 for humans, but this is not very important. The most important thing is that all this DNA is hosted inside the nucleus, which is very thin compared, very small compared to this whole amount of 2 meters, which is about 10 microns. So it's very tight compared to this length, which means like having 100 kilometers of rope inside the backpack. And this is very, it's okay, this is sort of astronomical analogy. It's like the distance between the sun, center of the sun and Uranus compared to the diameter of Earth approximately. So it's a very, large gap in time scale. And so how, this poses a big problem in how chromosomes are organized. And, but before understanding that, this is a very brief slide of what's going on inside, during the cell cycle. So for those who are not familiar with that, basically the cell cycle is divided in two parts. One is mitosis and the other is interphase. During mitosis, chromosome and they localize, actually, they are confined, each chromosome inside a very specific structure inside the nucleus, which is called the chromosome territory. So this was one of the major discovery in biology. This what I understand. It was mainly due to these two guys, Thomas Kremer and Christoph Kremer, two German brothers, who basically implemented this technique, which is called fluorescence in situ hybridization, to see chromosomes. So basically, we insert some fluorescent probes in the nucleus, the nucleus, and to very specific sections of chromosome, and you can see them. Now, the next technique, which was introduced before in the previous talk, is this high-C or chromosome or high-throughput chromosome conformation capture, which is a technique that reveals the amount of contacts, let's say, between the chromatin fiber. And also, this technique is, basically was confirmed somehow this territorial organization. So what we did at the time with Ralph was trying to understand basically a bit the physical origin of this structure. If they, somehow can be explained by just simple polymer consideration. And so what we did a very simple numerical experiment, basically using the standard Langevin molecular dynamics. And what we did was a very simple setup where we used the 4 model chromosomes, basically from 4 polymers, very long, arranged in a sort of mitotic-like conformation. And we used the molecular dynamic simulation to simulate the unfolding of these objects. So now these polymers are very big. So each one, so this is a zoom of this section. So each monomer here is just a very tiny amount of chromatin on the size of a 3,000 base pair, and each one is connected one after the other. Just like in a normal poly. So, and we let the simulation go. Very simple. Yes, we use the standard Kremer-Gress polymer model, and we took into account the very basic minimal ingredients, essentially the connectivity of the chain, some bending stiffness, the chromatin, let's say it's a bit like a worm-like chain polymer. We used we used the right amount of chromatin or DNA inside the nucleus of the cell. We did some mapping, and the most important ingredient was this one. So, namely, the hardcore repulsion between monomers. So, whenever these monomers move around, they can bump into each other, but they cannot cross each other. Okay. So if you do the simulation, yes. Yes. Okay. Yes. It's just purely repulsive. Yes. And yes, one monomer, because this is a coarse grain polymer model. So one monomer stays for about 3,000 pair of chromatin DNA. Which is about 30 nanometer in size. We use the radius of the monomer, right? Yeah, 30 nanometer. We assume that the 30 nanometer fibers come out, which is I know that people do not believe very much in vivo, but not don't believe at all. But yeah, that was the essence. But this is not very important. This fine detail in the end. Okay. And then, okay, we let the simulation go. So eventually, what it happens that, I mean, after you wait enough time, you end up with something like this, which is, of course, is a much more disordered structure, but still retains somehow the initial identity of this point. And with that, we start looking more into the detail of this peculiar. Each color is a different polymer. So 1, 2, 3, 4, and 1, 2, 3, 4. It's the same polymer, just a bit more disordered because of the thermal agitation of the simulation. That's it, nothing really special.
It's part of the regular configuration which has this loop structure. That's what we do.
Yes, this is our initial conformation. Yes, because we wanted somehow in our idea at the time was to mimic the, let's say, the folding of a mitotic structure. Of course, this is a bit of a cartoon, although it was shown by some recent work, is not so. It's not so different from what you have in reality, but also it's a bit more disordered. But I mean, it's just where we started from. Okay, and then you end up with something.
This road that you showed, these are your.
Sorry, this one?
Yeah.
The Boston.
The road is like 1 second.
This is like a flower structure. Each
This is like a cross section, and then it is a lot of helical structure.
You do these roads close to each other on the left side.
Yeah.
Each road stands for 1 segment.
No, stands for one, let's say, one model chromosome. So they do not cross, they are just say randomly arranged. It's like a road.
But in the previous slide or the next slide, when we show spring bead model, who is standing for the bead? That road?
No. A single bead is this one. So we have remember now, I think there's about 34,000 beads per single chain.
So
Yeah.
the question is that so this one chain is.
Yes, this is one. Yeah.
And going along.
Yeah, exactly. So you start, I don't know, here, for instance, with Mamoma, and you go there and you continue. So this is like a cross section, which is some kind of rosette, if you want. And then it continues along the axis. Okay, it's like an helix with a let's say non-conventional cross section the shape. Yeah.
And the scale in the bottom picture and the other picture are different.
The scales are compatible in the sense that so this is the box of your simulation because we simulate periodic boundary condition, and but the box is allowed to fluctuate but maintain. So we fix the external pressure, so we maintain the box maintains its overall volume, but it can change the form, can change the shape. These are roughly compatible inside the set. It's on the same scale. This is a magnification of let's say of this cross section of the visualization here. And this 700 nanometers is realistic in the sense that it's more or less the cross section of a typical mitotic human chromosome. That should be in this side. Okay. And the length of this polymer or model chromosome, if you want, is compatible with one human chromosome, one mammalian chromosome. Mice would not be different. Okay. Let's say it's one single human chromosome is about 100 million base pair, and this is about 100 million. I mean, the marking is about for 100 million base pair. Is it clear?
But you started with those rows and then you get this mess.
Yes, because then this is the initial conformation. Then we then we implemented Brownian dynamics. So basically it's a dynamics where you have some friction because this work is sort of implicit solvent. So this mimics the interaction between each monomer and the solvent. You have the Newtonian forces, which is some force field for between monomers. So it's the gradient of the potential. Then you have the noise, and the noise again is for mimicking the presence of the random interaction with the solvent. Okay, and then you put all this initial information inside your favorite molecular dynamics simulator, and you let it go.
And V contains those.
V contains all the full interaction for the between monomers, and this is just so you have this model. It's called PINE. It's a bit, looks a bit exotic, but in reality it's just a nonlinear spring. You implement this because you don't want the two monomers to go too far from each other because you don't want strand crossing. Okay, so it's a standard tool from this Kramer-West model. And then you have this potential that implements the bending stiffness of the chain because chromatin is not completely flexible, but I mean, there's some persistence length, which is, by the way, it's still a local scale. So it's I mean, in this scale, it's about 4 or 5 monomers. So it's not, it's not never super steep, let's say. Then this number here is in order to compute how large your simulation box has to be, let's say so, but that's easy to compute because then when you make an estimate of the right chromatin density inside the nucleus of the cell, so you can choose your simulation box accordingly, because this is with periodic boundary conditions, so you're not simulating a confined polymer, simulating polymers like in bulk condition, okay? So since you have periodic boundary condition, a monomer here will interact with eventually this periodic image here, okay? This is to avoid, let's say, final side effect. And then you have an article repassion between monomers. You mentioned this channel or whatever. It's basically truncated Lennard-Jones before the attractive interaction show up here. Okay, it's very, very simple, very standard. Great, and that's it. Okay, so we don't have anything else. So we end up with this, as you said, with this mess, which is actually a mess. So actually, but this, if you want, it's a mess, but still you have, I mean, after with some time of your simulation, you have maintained some sort of identity of your chromosomes. Something of course is just qualitative. So we have to understand this structure a bit more. So what we did at the time was to measure different different properties that can be compared to specific experiments, and namely experiments like we consider where this fish experiment, because by fish you can measure. So if you have let's say chromosome or a chromatin fiber, you can tag loci like this, and you can measure their. I mean, as a function of the genomic separation, you can measure the spatial distance between them, like you would do for a polymer if you want. No? But on top of that, you can also measure, again, for the same set of loci, how frequently they interact, and this will be by this chromosome conformation capture technique, I see, while this is by fish. Okay, that were the two, let's say, that were the two techniques that I compared to. So, and what we did, so we measured the mean square spatial distances. So it is a mistake here, it's mean square spatial distance between the two loci and compared to fish experiment. And we measure then the contact frequency, the contact probability, contact frequency between the same pairs of loci as a function of the genomic separation between them. And this is the result. So the symbols here are experiment from different, from available, because it's actually fish is a quite demanding technique. I mean, at least available at the time. So these are experiments, the symbols, for different organisms. They were yeast, fruit fly, and 2 different regions on the human chromosome 4, compared to the results of our simulations. In the lines are the results of the simulation, and as you can see that basically we were able to capture fundamentally the behavior of the result. And well, no, you just change the organ. So you have so this is the fish. So what you need to do, you need to prepare quite I mean, with a lot of work, specific probes that have to mark specific sequences. Okay, and the only difference between these different organs is the length of their chromosome. So this, the Saccharomyces cerevisiae, the ordinary here is an eukaryote, so it's somehow it's like us. but it has very small chromosomes. So each chromosome of yeast is about 300,000 base pair. So it's short compared to the human, or compared even to the Drosophila. Drosophila is a bit larger, it's 10 million base pair roughly. And humans are mammals are similar between them. Let's say it's on the order of 100 million. So it's 100 million more. So what the only difference here is the size of your chromosome. Okay. And so what we did here, we did different setups, but with polymers of different sizes, and a bit slightly bit different initial condition, but it's a bit of detail we can discuss later. Essentially, what just essentially the length, exactly. And what we found basically we were able to reproduce this data without any additional ingredient than it was the ones that were mentioned before. And and in particular, we were able to see this power law here that I will try to come back to this power law here later. Another thing that you can compare was by using this exactly the same set of simulation. I didn't change simulation, I just measure different thing. Is the contact frequency between loci, and again we were able to reproduce the behavior. And in particular, the contact frequency between different loci goes down as a power law, which is not surprising because it's a polymer, but it goes down as a very specific power law, which is 1/n. So I want to put the attention here that this power law here and this power law here are different from what you would expect from, just say, from random polymer, because for random polymer, you would see this is mean squared partial distance, so this should be proportional to L because it's just a random walk, or eventually a self-avoiding walk, a bit larger, but not 2/3. 2/3 means that the polymer is compact, okay? And this is also a reflex, if you want, of this power law here for the contact frequency that go down as L-1. For a random polymer would be this contact frequency would be much more repressed, would be like L to the 1.5, or for a self-avoiding walk will be even down. So these two power laws are very specific and very non-conventional for polymers. Okay. Yes. Yes. No, sorry, no. Yeah, sorry, this is I finished my paintbrush. Yes.
So just so I guess mapping experiments, it doesn't really care that your simulation is not in program, right?
Yes, that's a good point. Yes, in fact, this is yes. In fact, this is the next point. So we did also some mapping to time in some way, but this is not very. Basically, this showed that we were able to simulate some. In reasonable time scale. Okay, but this is not very important at the moment. For the time being. So, in fact, precisely as you pointed out, the fact that our structure are never in equilibrium is in the following sense. So if you start from something which is, let's say, polymers, which are very well separated from each other in some, let's say, in some box, and then you allow them to equilibrate, okay? Now, what it happens is that these things, of course, just by thermal motion will become disordered. But there are actually two mechanisms toward the relaxation, so which are somehow very well separated in time. So one is that you have some partial local relaxation, okay, but at some point this polymer will just bump into each other and will just get stuck because of this topological impediment that is in place. So eventually, very long time, they should do like this. They should mix, okay. But before mixing, that will takes a lot of time. And in fact, we estimated a bit this time how it should be if you take into account basically the only, let's say, generic reputation that we have in normal polymer formulated by Duchenne and Edwards a long time ago. And then you can estimate what would be the relaxation time of chromosome, and you can, and by doing some polymer-based argument, we show that for Saccharomyces cerevisiae, because chromosomes are very small, that would be very fast, let's say in some human times, but for very big chromosomes, that would take too much, okay? And so basically our understanding of the formation of this territory is because basically chromosomes always stay out of equilibrium. So this is a bit disappointing if you want, because then you cannot really say too much about that, because if it's out of equilibrium, then depends on the initial conformation. However,
Reassuring.
sorry,
Reassuring as well.
or reassuring as well. Actually, there was a paper that came out this year, because but it was really too biased, so I didn't understand that too much. But it was about the how chromosome territories age during in neurons, because neurons they never replicate, so that we have the same neurons that we have when we were born, and during the age they, and they made high C, if I remember correct, high C measurement on these neurons. They saw that some sort of I mean, my guess was some sort of degradation of the territorial. So it was quite intriguing, but I could not understand that too much because it was really biased. So I want to, so I cannot tell too much about that. But it was, I mean, to me, looked a bit. It was not related. I don't know if it was related to this, but it was quite intriguing. So anyway, if so, because of course, I mean, chromosome have ends. Okay, but since, as I said, I mean, these ends probably are not very important. Then we can neglect those. And we can imagine that somehow, because they're still all very well separated, we can somehow understand or try to understand the physics of this object as the way it was the physics of long ring polymers, which are unconcatenated and unconcatenated, sorry, and unknotted at all times. So that was our guess here. So now the idea is to make the same experiment that we have here, but for a method of rings, and to let them equilibrate. And the results was shown already in the same work. And so now if you do these experiments and you measure the same things, the mean square internal distances between now ring polymers which have equilibrated, you get the same results of non-equilibrated linear polymers that were the model for our chromosome. This is the plot that shows that. So here we have different lines. These are for our, so maybe you cannot see that, but the cyan line is for our, let's say, linear chains out of equilibrium, while the colored lines are for ring polymers in equilibrium. And we see that the bulk behavior is the same. It's basically non-distinguishable, saying that the physics of rings maybe as something that, I mean, we can use for understanding the physics of chromosomes. And this is quite, and actually this is some, I mean, my next point now. So now the physics of rings is very fascinating, or unconcatenated and not rings in maths. It's very fascinating because it represents still and somehow, I mean, in many respects, a mystery from the point of view of polymer physics. And the reason, actually, this system have already been studied since many years, but they are, I mean, there are still issues with those. And let's say, and the reason is the following. So let's take now these two different systems: a method of unknotted and unconcatenated rings versus a method of linear chains. So method of linear chains, I mean, basically we know almost everything now. So let's say one the fundamental work is the work by Pierre-Gilles Lejeune, who demonstrated demonstrated a few things, but I mean, one thing that demonstrated that the equilibrium, let's say, first of all, assuming that the linear chains in math behave like random walk, it showed that whenever a chain moves in, let's say, in an array of fixed obstacles that mimic the presence of the other chains in the math, now you should expect an equilibration timescale which goes like the cube of the length scale of the chain. So this is the main results of the repetition mechanism. But if you have a method of rings and you try to, let's say, to replicate a bit the same argument, then what it was found, I mean, what it was somehow hypothesized by Cocklock, Scheibe, and Rubinstein, was that the chain, what it wants to do in order to maximize entropy is to start to protrude through these obstacles. The obstacles here, I remind you, are just like the Gedanken experiments, mimic the presence of the other chain. Because the chain has to remain unlinked with the others, so it has to move through these obstacles and form branches. So the idea of these people was that this is a way to basically minimize the, let's say, the extent the ring interacts with the other ring, and at the same time maximize its entropy. Okay. And if you do that, it's possible to show, and it was shown in, especially by Shura Grosberg and by Jan Shura at the same time, and here it's a review with them on this topic. That let's say the ring has to assume a compact conformation. So you see now, let's say the typical size of a ring has to grow like the cube of its contour length, pretty much like the chromosomes do if they were compact. At the same time, you have also an equilibration time for the object, which is let's say, which is not exponent, which is very smaller than 3, it should be like 2.5, 2.6, at least theoretically. Let's say that's a theoretical prediction, but this is still a bit debated. So on this, there are still controversies. Yes.
The persistence length is of the same order as the length.
Yeah, here persistence length is irrelevant in the sense that here I'm assuming that let's say the distance between the obstacles, that again is the mesh size of your solution, is let's say the same size of the persistence length or even a bit larger. Okay, it's larger, yeah, or larger. So it's persistence length here is not important. Okay, so I'm talking about the large scale behavior. A bit like the same thing that you have in melt of linear chains. In melt of linear chains, the tube, let's say the rotation model or the tube model of the Jen and Brian Edwards is always above the persistence length of the polymer. So it's exactly the same ideas here. It's a very coarse model.
Biologically, that's right. I mean, if finite, then it might matter.
Sorry?
So biologically,
yes.
This is true.
What would you mean?
So I know in these limits, you don't have a persistence there.
Mm-hmm.
I mean,
If you mean the persistence level of the chromatin,
yeah.
Well, for sure, for sure, it plays a role whenever you have. I mean, I assume, for instance, we have seen before this promoter enhancer interaction. If they are relatively close along the sequence, for sure, it will play a role. And here I'm talking about on a very coarse vision of the model. And of course, I have to completely negate one more thing. Here I'm considering only homopolymer. Of course, chromatin is not an homopolymer. There are already now many studies to show that the sequence is, of course, super important. Just a standard persistent length, but I mean, what I'm talking here is always something that goes beyond that. Okay, on very large scale.
The question is mesh size. You have to have an mesh size.
Yes,
And mesh size is much larger.
in this case, no, not at all. It's a bit 4 times the persistence length. It's actually in the so if you do the calculation, you estimate to do the proper mapping, we saw that this is about what's called loosely, it's in the realm of loosely entangled polymer. So it's the entanglement length for this object is roughly about 4 times the full segment. It's a bit larger, but compatible. It's not 20 times. Okay. And yes, yes, and this is also because if you do the calculation, we'll see essentially that basically these polymers are like in a very, semi-dilute solution, so it's not really a proper melt. So we're talking about also above the entanglement length of the solution. Okay, so yeah, thanks for the question. It's quite important. So now, can we? Okay. And here is a work that I was mentioning this model by let's say this randomly branching polymer model for rings in a melt by Cochran and Shire by Rubinstein. So the idea is that again, if you so the structure is like having your ring that to minimize the extent of interaction toward the other rings that are running them, at the same time maximizing the entropy. And so the idea is that this thing has to do some double folding around a tree-like structure. So the next step will be with RAP was precisely to implement such a kind of model. What we did, we basically had a model of branching polymer around those each one, around each one of them, we double fold the structure of the polymer, and we compare to brute force dynamical simulator. We're able to construct now all bunch of conformations for ring polymers at equilibrium, and we saw that this model somehow describes the physics of rings in ML. This was a bit the background. So, however, and this is a bit now some of the open problems in, let's say, in the realm of ring polymers, is the fact that let's say once in a while you have things like that. So imagine that so for linear chains, you have always that each chain is protruding to the others and moving, but essentially without the topological constraints are kind of non-permanent. So they evolve in time, but they are non-permanent. Well, for rings, this let's say unlinkedness it's pretty important, and this is permanent by construction, if you want. However, because of let's say of the ring topology, nothing prevents you to have some sort of, let's say, the loop can open, okay, and can produce this kind of structure here. And you can have many of them. So each ring can allow some opening and produce all this kind of structure. So now it's something which is now, let's say, of interest for many people in polymer physics. So this kind of structure, which are named threadings, in the community, have a lot of important consequences in ring system. And that was a bunch of applications, and there are others. And so this was the first that came to my mind in on the subject, where these threadings become important for rings in extensional flow. And that was a group that saw the involvement, I think, of Michael Rubinstein and also Gary Grest and other people. Basically, you see that when you have a ring melt under extensional flow, these are called stretch, and once in a while you produce some sort of this conformation, and then they start to be, let's say, not in the usual sense, but let's say they produce such kind of structure like a tie, and they stretch. Under this condition, you see that this gives rise to a non-conventional elastic response of the medium. In a work which was done now a few years ago by Davide Michelet and Matthew Turner, they showed that was something very, very, very nice. So they have, let's say, an ordinary ring melt. Then they froze part of the population of this ring melt, and they show that basically the system gets stuck, so it doesn't move at all, like or removes very, very slowly, like in a sort of glass. While if you do the same experiment on linear chains, you get some, it becomes a bit slower, but it never completely blocks. So this seems to really depend on topology of this structure. And very recently this year, it appeared another work, it's an experimental work supplemented in my molecular dynamics simulation, where they measure the elastic response of these rings. And while theoretical considerations seem to suggest that basically the system is always relaxing and relaxing with the power law behavior in the stress modulus, instead at some point, you should see some behavior which is more like similar to the one of remember the one of linear chain. And this behavior here was described to the, let's say, to the formation of these threadings. Although I would like to underline the fact that all these kind of experiments were done by really stressing the system out of equilibrium, while the presence of these threadings, let's say in a normal equilibrium situation, it's still to be understood. But just to mention, I wanted to mention this just because I want to show you that this is an open problem now, where people are actively working on that on the community of polymer physics. And there was also a workshop this year in Italy dedicated to it. Okay, this is something that we had to do with Trebis, but I want to talk about that. I want to move to the next topic. So I don't know if you have a question on this. Yes. Yes.
Yes.
Like, you mentioned a component that so it may indicate, that this can come on this kind of equilibrium.
If I make chromatin in equilibrium.
Yeah.
So if I, from the point of view, that would be just like a spaghetti ball.
Always,
Yes, yeah, for sure, it's out of equilibrium in some sense.
can make it like out of the dynamic. I don't know if I got it here, maybe.
I see what you mean. Well,
But
the,
then,
good for the cell, you mean for us, you mean?
Yeah.
Well, there are, the problem that there are different, well, if you are out of equilibrium, there are different ways to being out of equilibrium, okay? So the point of this, what I present here, more to show, let's say, a physical approach by which you can describe, let's say, large-scale behavior of chromosome without too many essential ingredients, okay? For instance, now you can ask, you can ask how we go back in the sense that because now this thing it's a sort of a mess that was before, but now you can ask how you can go back to mitotic chromosome, and this is not something that I mean can be explained by this kind of model. So there are now many results about a specific active mechanism that act on the chromatin fiber and then recompact it again. Okay, so for instance, this is also an out of equilibrium process. But it's two different stories. And there are, I mean, many more other interactions that are in place in chromosome. Okay. That was just to show some simple physical model. There's connection also to the realm of ring point.
One more question.
Yes.
What's
The knots, that's a good question. The knots, I think it's because the polymers are too big and too large. It's possible at the very end you get some knotting, but it's not important.
In the end, can never find each other.
No, okay, they can maybe one end can find itself and locally become knotted, but this will not be observable. I think it will be very, very rare to have some kind of. I didn't measure them to be honest, because I can't be sure, but I think it's not really important. Yes.
Might be a really random question, but I'm thinking it's
Mhm.
get these. Like, would it adopt ring like?
So each loop is a ring.
Yeah.
Yeah, it's possible. Yeah, but this is sort of next detail on this framework. Of course, you can do. I mean, you can study all those sort of things. And the question is how that will change the overall statistics. It's possible that it will change. This I don't know. For sure, it will change a little bit, or it will change maybe a lot. I don't know. I simply don't know. But it's a good question. Yes, I agree. It's a this I can tell. I don't know, but it's possible that it will change the thing.
Because I thought it's regarding fish and sea. So when the chromosome that is used is it what stage the life cycle is it in? And does it matter?
You can do fish or IC in different during different life conditions. So the very yes, it changes a lot actually, because when you I can show you a paper. I don't have here in the slides. Actually, the same persons that I'm talking about now high C, because I think that so the same person, at least a group of same person that implemented high C originally for interface chromosome, they made high C also for mitotic chromosomes, and they show let's say how the because I see summarized also by this contact matrix. We have seen an example before here also here, and they saw how the evolution of contact the evolution of contacts progresses in by doing this IC, and it changes a lot actually. Basically, during mitotic phase, the IC matrix is boring. You have only the center line, which is basically due only to the fact that you have a polymer. So of course, contacts which are close in sequence are also close in space. So you have a very thick line, and then you have noise, if I'm correct, or no structure. While when you enter into interface, you start to see the formation of structures, because IC matrices are not really random. So you have a sort of plate pattern, while maybe I can show you. It's a bit visible in my first slide. Yes, here. Okay, these are the really first IC matrices. Now you have much better result. Because this is the resolution of 100K. So each is like a pixel matrix. Each pixel is 100K. You have now better resolution, but the idea, let's say, but it's the same. So this is let's say the old, or roughly the old chromosome for human chromosome 14 length. So this is as you can see here, this is in IC during interface. I think it's during G1, so it's very the longest part of interface. And as you can see here, the structure is not random, so it's not like what you would expect from an ordinary polymer, especially because these are ensemble measurements. It's not one cell. One cell, you can say, okay, it's one cell. It's one single realization. No, this is really an average of between thousands of cells. And you can see here you have structure for a polymer or polymer. So there are for sure specific interactions. These are also being demonstrated by other people. And the work, I mean, if you work a little bit, basically you do principal component analysis of these matrices, you will, I mean, here what they saw is that you can partition the chromatin into two, let's say, into two subspecies, if you want, they call it A and B. And let's say the A and the B interact more, while A and B interact less. It's a bit what is shown in this plate pattern color. So you see a lot. Why exactly during the mitosis you don't see that. You see something very flat, apart from the diagonal, which is just because let's say they are close in sequence, so it's something trivial. Okay, and then you have also some substructure which were discovered by a group of Bing Ren in San Diego, where actually, if you go deeper into this square, you see, let's say, a more refined square, and so basically regions inside one of these large domains, which interact more and but less between themselves. And they call these things topologically associated domains, and they're well, to some extent they demonstrate their function or all these kind of things. Then they are maintained by specific proteins, so you can show that if you disrupt this protein, then so it changes a lot. But the thing to have in mind is that both IC and fish are static. Technique, so basically you build the cell, or sort of. I mean, you have a really a picture of them, but it's not dynamic. Otherwise, you have to use other technique to see the dynamics of the cell. Okay, so this is for the first part. I think I don't know much time. 10 minutes, not much, but it's okay. But it's good. You know, we talk a lot, so it's nice. I like it. So this well, I don't know how much time I have. So this is just some recent work. It's about an RNA. It was done in collaboration with some people in Ljubljana, with my friend Luca in Trento, with Rudy Podgornik, now in China. Rudy was before a professor in Ljubljana in the same Josef Stefan Institute. So the idea here is to basically to approach to study basically RNA molecules by using the language of randomly branching polymer. So what the two things have to, I mean, what the two things have in common. So first of all, one has to need to understand the physics of randomly branching polymer. As I said, I mean, I started this thing because the rings, somehow we have like randomly branching polymer. Now, so forget about the rings. Let's consider only the branch structure. So branching polymers are interesting. I mean, because they are classical systems in polymer physics, and also because I mean their physics is much richer than ordinary linear polymers. So now, in a linear polymers, you have only one exponent, the exponent nu, that describes basically how the typical polymer size grows. For a branching polymer, you have actually one, two, three, four, at least three more exponents. Huh? So one exponent is the exponent rho. The exponent rho describes how the typical path length on a branching polymer grows number of monomer n. If just for a polymer, this rho is just one, ideally. For a branching polymer, will be something different from one. There is another exponent which is called epsilon. The exponent epsilon describes how the typical, let's say, branch size of your, let's say, branch polymer grows the number of. And basically, to compute that, you randomly cut one bond and you measure the size of the two branches. You take the average and you get the sufficient. Actually, it was demonstrated now almost 3 years ago by Prof. and Madras. These two exponents, epsilon and rho, should be equal. Although they are very different in nature. Okay. And then you have a fourth exponent, which is less important because it's a combination of the other two, which gives you the typical mean square spatial size of a path as a function of the path length. Okay, and by just scaling consideration, the new path, which is the new exponent, depends on whatever. Basically, new path has to be just the ratio of the two. Now, how we understand these these systems can be understood by using some generalized fluid theory. I mean, fluid theory is not, we know that it's not exact, but it's, I mean, but it gives you more or less the right physics of the system. And actually, the main contribution came from Isaacson, Lubensky, and Rosberg and collaborator. Basically, they show that the free energy of let's say branching ensemble contains 3 terms instead of the 2 terms of the classical fluid theory. So one is the classical elastic contribution that comes from stretching your polymer. One is due to the entropy of branching, because here branching can rearrange, so they are annealed, so they can rearrange everywhere in the system, plus the ordinary excluded volume. Okay. Now, the interesting thing here is that, okay, this is a bit phenomenological, but let's say the interesting thing here is that these 2 terms, okay, they do not depend. So sorry, this sorry, let's say the other way around. The excluded volume term does not depend on n, typical path length. This means that these 2 terms can be, just by using the usual Flory argument, can be minimized without taking care of this, and this provides a relationship, a mechanical relationship between rho and nu. Okay, that should hold whenever, I mean, whatever it is the ensemble you are considering. And now, this is a collection of, let's say, of different randomly branching polymer in different ensembles. So we do have cover. So we have randomly branching polymer, single randomly branching polymer in good solving condition, in theta solving condition, and in a melt. And this is a collection of all these exponents that we recapitulated in this review. And you can now, let's say, compare this mechanical relationship with, let's say, to this measured exponent. And we found a very good agreement between the measured rho and the measured nu with this, let's say, phenomenological relationship within error bar. So this is non-trivial because this comes from Flory theory, which is very phenomenological, very, let's say, approximate, but still gives us, let's say, a good way to understand these results quantitatively. So now what does, okay, then, I don't know if I have time for this. Then there is more. So there is a, I mean, you can study also distribution function. This is useful for what we want to do for RNA. So you can measure also the distribution function of these different quantities, and you can show that these quantities, they all obey a very specific, let's say, non-trivial distribution function that were quantized by the Croiseau, Pincus, and all these sort of big guys in polymer physics, that you can find some scaling relationship between all this exponent and the corresponding distribution function. And okay, I want to skip this. Well, it's a bit too long, but I want to go to this. I mean, to idea. Then probably I will stop. So if you are curious, we can discuss later. So what we did here, we did a study on the branching nature of RNA. So why RNA is branching? So an RNA forms all this kind of structure just because of the base pairing. Okay, and I mean, it was shown by now in many works that actually, when you compute or when you estimate, let's say, the secondary structure of RNA, it produces a lot of these structure. In particular, produces a lot of multi-loops from where you have the different pair strands protrude from. And in particular, so this is a very small RNA molecules, but if you consider these RNA molecules of which is very large, relatively very large, of the dengue virus, you can see that forms all sorts of this structure. So the question we asked is that can we use, let's say, can we use the physics of branching polymer to understand, I mean, let's say, the scaling behavior of these RNA molecules? That was overall our idea. And I mean, why? Because it was shown that some degree, I mean, the right amount of branching was used in the virus. I mean, sorry, in the RNA of some, especially of viruses, to let's say, to force the RNA, the RNA molecule to inside the corresponding capsid. Okay, so this is an example of some different viruses that, according to the shape of the capsid, you have more or less a degree of branching. Okay, so what we did here. Okay, the inspiration was okay. Here with this I stopped. And if we want, we can discuss a bit more on my specific results. So the idea here is was also motivated by some more or less recent work by the group of Bill Gelbart in the United States. What they did, they precisely the same thing. So what they computed, let's say, the branch. So let's say they compute the branch structure of many viruses and also of, let's say, artificial RNA molecules. And what they show there that let's say they introduced the following quantity, which is the max, what they call the maximum ladder distance. In my language, would be, let's say, the maximum path length on this, let's say, on the obtained branch structure, as a function of the total RNA size. I mean, in terms of base pair. And what they show is that if you compare the results, so the results for random RNA, let's say for RNA which doesn't exist in nature, but it's just an experiment you can do numerically, the typical size of this maximum ladder distance for random RNA is grows with a power law of about 2/3 as a function of the total size of the RNA. Vice versa, viral RNA is much more compact than that. So here is the result of the random RNA. Here is the result of a viral RNA, which behaves completely different from the random one. And they asked why. The interesting thing for us was also to have an understanding of this exponent here, which is what we did recently. And because of the connection between the exponent nu and rho, because this is actually the exponent rho, no, that I mentioned at the very beginning, and because of the connection between rho and nu, have an understanding of the exponent nu in random RNA, and we arrived at different answer than what the group of Gelbart did. And so what we did, we introduced a pipeline. We have analyzed hundreds and hundreds of RNA structure in order to have a complete understanding of the statistics of RNA molecules and make let's say, and determine all these different exponents that to understand the molecules. And but I think that my time is a bit over. So if you're curious, I'm happy to talk to you about this because it's a very new work and we have ideas a bit how to move further. So with this, I think I will stop. Thank you. I don't know if you have questions. Yes.
Physically or biologically relevant, but if I have ring polymers
Mhm.
of the mouth,
Mhm.
maybe they kind of meeting into each other. Now, if I, from a random direction, if I start to pull on one, there's going to be like a shoelace. There's going to be one particular direction that everything's going to unravel.
Yes, this is a very good question. I think
So for linear chains,
yes.
well, I should pick, but actually for loop structures, it might be some.
Yeah, I don't know if this answers your question, but there were in the end, I don't think it's very different what they did there. So there is the work by Jan Sandbeck. What they did, I don't know if it answers your question, but let's say I think it was a bit similar SPL. What they did, they had their mat, actually a real mat, so something not my I am in semi-dilute solution. They have a real mat, and what they did, they just for one ring. They create, so basically they use molecular dynamic circulation, but with where they use monomers on single ring with a different temperature. Okay, so what they did, this basically drives again the system out of equilibrium. So they saw that sometime, I mean, at some point, this thing gets stuck too. So when it's basically, so you really see in the moves, these things start to being pulled out, but then it becomes blocked because of the presence of these threads. So they show quite clear this because of that. Yes, yes, yeah. Yes, when you pull, yes, it's the same thing. Yeah. Yeah.
Thinking of spaghetti and pulling that over.
Yes.
Thinking about that reaction of that.
It's a bit the same. It's a bit the same problem, I think. So because whenever you, I mean, what I see, whenever you drive, let's say, if the rings are long enough and it's in a very, let's say, dense condition, whenever you drive them out of equilibrium, you observe the effect of this thing. Well, if they matter also, I mean, in equilibrium the static situation, I'm less sure because I mean, probably this thing do not matter to this threading do not matter too much. Actually, we made a computational study where we basically changed the standard of threadings by modulating the persistence length of the rings, and it seems that this we collaborated with Jan on that. So the threads exist, but they are on a very local scale. So it seems that if you want, I mean, from the static point of view, they do not matter too much. So it's possible, but of course, I mean, if you do such a kind of an experiment, like would be like a generalized microarchaeology, you probably would see something like that. I don't know.
The unraveling, mediated.
Yes, yeah.
That's essentially like.
Yes. But for that, in many of, for instance, in this, yeah, because it can get stuck some point. So in these works with by Leonid, yes, what they also to introduce some finite amount of let's say topo 2 or topo 2. So they had to allow some strand crossing at some point. Otherwise, the system probably would get really stuck. Yeah, of course. I mean, it would get stuck because they are very long polymers in them. Sure. You like interactions. This one, right?
Yeah.
This one?
Yeah.
So
what actually you are working with this agreement?
These ones.
Yeah. These are the neighboring B.
This one. So okay, maybe I don't know if it's. So this is this contains all the coordinates of the of my system. And because in general, I mean, my single monomer here can interact with all the others. Okay.
Then go back to the picture where you,
This one?
yeah,
Uh-huh.
so. Whatever you see.
V is it's a bit complicated, but V is a bit basically is the sum of all of that. So if you want, this is the thin interaction that this is a bonding interaction. Okay, so one monomer versus okay, I see for many notation not to be here. Let's say this is a chain connectivity. So it's one monomer on the chain with its nearest neighbor along the chain. Okay. So this is the bending stiffness,
Yeah.
and this is let's say a 3 monomer interaction, but always on the same chain. Okay, this is the density. So while this is any pair between any pair of monomers. So because this is exclusive volume interaction.
So the different chain, all indirect,
Only exclusive volume, yes. It's all the same, no difference. It's all a completely boring system. Let's say.
because it's secret.
Well, it can be, no, it can be. Actually, this is a very nice topic. I mean, there are some of my some of people I know, some friends, they do precisely what you say. So namely, they have they don't have a homopolymer, which are really two polymers exciting. But I mean, they can tell you a lot about this. But let's say they have what they do. They because of so the idea was to a bit have an understanding of this plate pattern here.
Yeah.
Okay. So there are different ways you can obtain this plate pattern in polymer field. One idea is that because I mentioned before that according to this original of Decker, that chromatin is partitioned into 2 species, let's say, probably there are more, but let's say, I mean, in the intelligence scale there are only 2. They use a polymer model where you have portion of your chromatin or your polymer. They call A, and next portion they call B, and they interact only preferentially with each other. And they show that then what you have is some sort of microphase separation, like one, let's say, ordinary A and B system. And when you let's say you will measure your simulated, let's say, you measure your corresponding asymmetry with your polymer polymer, you obtain a plate pattern with this guy. Okay, but it's not the only possible mechanism. There can be others that can explain quite well with the plate pattern.
But you didn't do.
No. If I do this, if I compute this isomatics from my simulation, you will tell me different plot. You don't have any sequence information. But I could do that, of course. I mean, I just can introduce the same mechanism and I would obtain the same result. Okay, so when you measure these, when you measure, sorry, what is it? Yes, this is probably an important point. When you measure all these properties, which are the mean square endpoint distances or the mean conductivity as a function of genomic separation, you are always do some kind of averaging. Okay, because how you do that? For example, this is very easy to understand how you do that. So you have your I mean, now there are websites where you can download thousands of these markers, so you can check yourself. So what you do, because so basically here each diagonal means a specific. I mean, it means the same genomic separation. So to loci, okay. So you have one loci here and the other loci there. So on the diagonal means the same genomic separation. So you can average over all genomic separation. I mean, over the diagonal, which means taking the average of the entries. You are measuring the mean contact frequency between loci and the same genomic separation, regardless of the nature of the loci. So you have now an average contact. But it's what people do when they want to sort of get a signal from this line. Because the signal is very faint, it's not good, I mean, not an enormous signal. What you do, you take your what you get from the experiment, and then you average the observed versus the expected. The expected means the average signal, and you get some, you know, something that will become higher than the average signal or lower than the average signal. When you're higher, it's called, they call it, I think it's enrichment, means that the contacts you measure, or the contact you get gotten is something probably significant because it's above the average, let's say, of what comes from just the polymeric nature of the chromosome. Let's see.
So, to how to data point
It was. Sorry.
Yeah.
This one?
So, yeah, because this is actually a how do you decide when to actually stop it?
That's a good question. No, what we did here, so you have this kind of thing, then you wait, and some point this thing does not change anymore because it's. I mean, the equilibrium would be. You cannot simulate those. I mean, you have to wait forever. At some point, the thing gets stuck. Yeah, you can measure, well, you measure different things, you can measure duration values on this object, you will see that the force of the medium and get stuff. And then you average, then you can make the simulation longer, of course, but then you stop. What makes me believe that this thing is okay is because if we compare, as I said, sorry. Actually, you can see. So if you compare the bulk of the let's say non-equilibrium linear chain towards the bulk of now equilibrated rings, the behavior is the same. So it means that you have reached a state which is non-equilibrium from the point of the linear chain, but it's equilibrium for rings. Okay. Actually, but okay, but to be honest, let's say an indication the structure remains after equilibrium and that the memory of the initial conformation is here. These are for linear chain, and because they were constructed like let's say sort of cylinder, you have something that goes up. Yeah, but this is not saying nice in the not very constructed form. I mean, of course, it's the auto equilibrium because you know that we have constructed structure by hand, so you have.
You predicted power that was on the previous graph.
Yes, so this is the
end result.
this one you mean?
Yeah.
So so this actually is the one that you would get, this one, and also for the contact frequency. This is the one that would be predicted. So actually you can understand this power law by using the method of rings, because the method of rings, the method of rings are compact objects, and they will show a power law behavior for contact frequencies like of this one. So this is something that is now known, and this is how we understood this thing. It's not an analytical risk. Oh, well, it's a semi-analytical sense that there are more or less analytical speed. But actually, this and this are sorry. This one is a bit the consequence of this, because if you assume scaling for let's say any polymer, let's say provides big enough of some scaling relationship for instance for the mean square internal distances. So now just because they have you can say that so let's say that you have only one relevant scale. So let's say the distribution function of my endpoint distances, and it should be like a scaling function. So there should be something like that, And then because you have a Jacobian in front, this has to be normalized. It has to be something like, and you are in space. So this would be like, let's say D over what? It would be like D, because D is the measure of space 3. My 1/3 is one. So if you integrate this in over VR, because this to be that has to be let's say 1 constant. Okay, that's just the scaling, the ordinary scaling you have for polymers. For a random polymer, a random walk, you have the same thing, but not 1/3, but 1/2. Okay. So in fact, this is the okay. Here I have neglected the. I have neglected chain correlation, which are of course important, which is this exponent theta in the exponent polymer field. But it's has to be very, very small. Also for ordinary self-happening work is small, I think 0.1. So it makes correction to scaling, but it's not something that you can observe if you compare to experiments. For sure, experiments are much less. 1/L is as good as 1.1.
So on the one,
So I guess we don't continue this.
If you want, we can talk. I don't know. Oh, we have to. We have to leave. No, I don't.
Yeah.
Ah, yes, sure. Ah, because I have to stop here, right?
Thank you very much for.
Thank you for the nice question.